Thank you. All right, so it is my distinct pleasure to introduce today's keynote speaker, Lalita Ramakrishnan. I've known Lolly for a long time as colleagues and friends, and this is really truly a great honor and pleasure to be able to bring you back here to give this keynote address. And I figured I will give you a little bit of background on Lolly. She was born in Baroda, India, now called Vadodara. Okay, anyway, so I'm not going to get into local politics of that, but that's where she got her medical degree, and she started medical school when she was the age of 17, and then got interested in immunology through one of her classes, and then came to the United States to get her PhD, while at the same time doing her medical residence in New England. And she did her PhD in B-cell differentiation. And then along the way, also she was a resident there, and then moved to UCSF to do a fellowship in internal medicine and infectious diseases. And she has had a long interest in tuberculosis. I mean, this is something that has touched her directly. Her mother had several bouts of spinal tuberculosis when she was a child, and so living in India, she was exposed to the great impact that TB has on people. And so when she was deciding how to design her career as a scientist, she wanted to work on TB, and she wanted to go to one of the primal labs in microbial pathogenesis, which is Stanley Falco's lab. And so she approached him about doing postdoctoral training with him. And legend goes, something along the lines that there was no way in hell that he was going to work on an organism that took a month to grow on a colony, and that it only infected humans. But that she could come and work with him if she could find some alternative. And this is when she started working on this idea of finding a pair of pathogen-host interaction system to be able to develop in an experimentally tractable way. And this is how she ended up working in Microbial Pathogenesis, which at the time was known to infect cold-blooded animals, causes this disease called fish tank granuloma. And she developed the system on Microbacterium marinum infecting frogs. So that's how she started. So there's hundreds and hundreds of frogs that got injected with Microbacterium marinum to create both acute and chronic infections, and then she reactivated disease in these animals by immunosuppressing them, but apparently takes injecting drugs into them daily. So there was a naive graduate student who helped her with that, and then he got cured from any idea working on this organism. That was me. I wanted. So I actually have a paper with Lolly, and so I'm very proud of that. I have two papers with you. Okay. So this was the beginning. So Lolly established the system, established genetics in the organism, started the animal models. I think she always had this idea of using zebrafish as a system, now that she knew that she could get diseases in these animals. Oh, and by the way, she also published an article in New England Journal of Medicine where she proved, okay. So apparently it also can cause disease in humans. So I'm just gonna leave it at that. But at any rate, yeah, so it does cause disease. So she forever changed the landscape of how biosafety classes are given at Stanford, and so you left a long-lasting mark on that institution. But so then she took a position at the University of Washington, and she truly then took off with a zebrafish system, especially with zebrafish larvae, where she could image the process of infection in live animals and in real time, and made some seminal discoveries about the genetics of the host and how they impacted the outcome between the bacteria and the host, but also she was able to fully take advantage of the imaging capabilities in these transparent organisms to follow the formation of a granuloma essentially in real time. And there was an underappreciated dynamics to this system that had not been appreciated in tuberculosis. And so she had a series of landmark studies, one after the other, and the one that I thought was the most impressive is this idea using forward host genetics to identify susceptibility loci in zebrafish that then turned out to predict what its sensitivities will be in human populations, because she looked at cohorts in Vietnam and find out the mechanism by why some people were more susceptible to meningeal tuberculosis, and just go back and forth between observations in humans and in animal and the genetics of the host. Since then, she moved to Cambridge, where she is the head of the immunity group there, and she keeps she just keeps batting them one after the other. And the latest paper she has, that you can see in Cell, is about necrosis and TNF and the role of mitochondria and ROS and how that is controlled by Microbial marinum. Along the way, of course, she's got all kinds of accolades. So she sent me her CV, and I'm not gonna.
He asked.
I asked. So we'll start on page 67, where we list a lot of the. She's been elected to the Royal Society. She's a member of the the Academy of Sciences. She's a member of the EMBO. She's a microbiology fellow. She was a recipient of NIH Merit Award. Too many to mention. And I would like to finish this and on a personal note, and also because I'm sick and I'm feeling like whatever I took is about to kick in any minute. And I might as well quit while I'm ahead and they're filming. That Lolly has just been an outstanding mentor of scientists along her entire career. So not only people who work in her lab, but everybody who came in contact with her is talking science with her is fun. You give advice both of personal nature, scientific nature, and every time I come to a meeting and I know you're going to be here, I always look forward to it. So with that, I'm going to leave it to you, and we look forward to your talk.
Can you hear me? Thank you, Rafael. Do you have any questions? All right. It's really a great honor to be doing this keynote talk. I've been coming to this meeting for many years. I've organized the meeting. I've done the 3-cycle thing as an organizer. And I might slightly owe my job to this meeting because when I was a finishing postdoc in Stan's lab, finishing in Stan's view, but not necessarily in my own view because I was pretty happy there, he presented my work at a keynote meeting, at a keynote talk at this meeting in the early days of its inception, and he spoke about my work. And shortly after that, I got a phone call from the University of Washington saying, hey, you know, we've got this job and the application's closing in 2 days. Can you please fax us? Those were the days of fax. Can you fax us your materials? So I did, and so it's been great. So thank you again for inviting me. Okay, so I will tell you what remains of my story to be told after you've heard Rafael's introduction. And yeah, thank you. So I'm going to tell you a story today about the host genetic susceptibility to TB and what it's taught us about pathogenesis, what it's taught us about basic biology, and what it's taught us about the potential of getting new drugs for TB. And the work was initiated in my lab by David Tobin when he was a postdoc, and he's now a professor at Duke, and then picked up by Francisco Roca, who's finishing up his postdoc in my lab. So let me just, yeah. So we all know that TB remains a huge problem, has 10 to 11 million cases a year, nearly 2 million deaths a year, and that's despite us having more than about 60 to 70 years of chemotherapy. The first drug, streptomycin, came out in 1948. But the point I want to make from this statistic, this very dire statistic, is that while on the backdrop of this is this somewhat hidden fact, a fact that's unsurprising, that the susceptibility to TB varies a great deal even among so-called immunocompetent individuals. So everybody who apparently is normal will respond very differently to TB. And this has been known for a long time through long-term epidemiological studies that unfortunately sometimes get a little bit forgotten, but is nowhere better highlighted than in this very sad experiment that got done inadvertently. And this is called the Lubeck disaster. And what happened was that when BCG first came out, it was a live attenuated vaccine. And when it was first developed in 1921, an effort was made to roll out BCG and inoculate everybody with BCG systematically. And in the north of Germany, in the far north, which is, on the surface thought to be genetically homogeneous, so certainly there were no immigrants or anything like that back then, infants were given BCG vaccine. And it's always given to newborns, and then it's given at a monthly dose, 2 more doses. Unfortunately, the vials got mixed up, and what they got given was virulent tuberculosis. So infants got exactly the same dose of virulent tuberculosis at the same time, as in hours after birth. And yet, the outcomes were very different. 72 of the 251 died, so that's roughly a third. And then the remaining 2/3 showed some sign of disease, either radiologically or possibly some lymph nodes popping up. But of them, half had overt disease and recovered, and the other half didn't have any disease at all. So in other words, a third died, a third got sick but cleared the disease, and a third never got the disease. So what could be the basis of this? And if you think about it and consider the lifestyle of TB, it's not a big surprise, because it's as with many bacteria, a complex lifestyle. So TB is aerosolized in the cough. Raphael, yes, thank you. In the cough of individuals who are infected, and the bacteria coughed up in fine aerosols. They're inhaled by the unfortunate individual who happens to be near them, and then they get taken up by alveolar macrophages or possibly other kinds of initial sentry types of cells, and then they get in through these cells into the body, traversing epithelia, lung epithelia, and they now have learned to subvert killing defenses of the macrophage, and to basically subvert endocytic trafficking in the macrophage, and have learned to grow in them, and to then cause the macrophages to form these strange structures called granulomas or tubercles. And within these granulomas, the bacteria can grow even more. And then there's a final step that's quite important, and that is that the center of the granuloma has to undergo what is called caseation necrosis, where the macrophages have to undergo a necrotic death, where they pop open, release the bacteria, and it is this gemisch that is then coughed up, and it is the necrotic granuloma that's essential for transmission of the disease, and therefore for the evolutionary survival of this bacterium, which has been surviving for millennia. So Raphael alluded to the fact that I convinced Stanley to let me work on TB if I could find a surrogate model. And the organism I found was Mycobacterium marinum, which turns out now on sequencing to be the closest genetic relative of the human TB bacteria, but there were already hints that that might have been the case when I decided to work on it. And Mycobacterium marinum was found because it was killing fish in this aquarium, the Philadelphia aquarium, and people didn't know why the fish were dying. When they autopsied them, they found tubercles. They found the exact same granulomas that we see in humans, and they could see the bacteria in them with the special stains that are used for TB. But when they tried to culture them, they couldn't grow anything. And until Aronson had the brilliant idea that fish are cold-blooded, and so maybe they should try to culture them at low temperatures, and when they did that, they could grow the bacteria, and that's how the bacterium was discovered. Well, it turns out that Mycobacterium doesn't just infect fish, it also infects the cooler surfaces of humans or any warm-blooded host. And I know Raphael thought that this was kind of, I mean, he mocked me for this, but I will tell you that this came in handy for a publication in a very lean period of my life. So having demonstrated this, I went back and looked at various of its natural hosts, ectothermal hosts, and eventually I settled on after torturing Raphael for many years, I settled on the zebrafish. Now the zebrafish is well known to get disease from Mycobacterium marinum. People who use zebrafish for various purposes dread this disease. And here's an H&E, a hematoxylin eosine stain of a human granuloma, and you can see it's nicely organized, and you can see that there's that necrotic area in the middle, as evidenced by the fact that you don't see any blue nuclei there. And here is a zebrafish granuloma that I've stained with that special stain that stains bacteria, and you can see that it's nicely organized. There's cellular areas that have a few bacteria in them, and then you can see that the bacteria are growing like gangbusters when they've lost lost the controlling influence of the macrophages and are growing extracellularly. But the real power of the zebrafish is the fact that it's transparent for the first 2 to 3 weeks of life. And so we wanted to see if it was possible to look at infection in these larval fish. And indeed, we could see single macrophages that were infected with the bacteria, as well as aggregated macrophages. And when we engineered a fish to have green fluorescent macrophages and red fluorescent neutrophils, infected them with blue fluorescent bacteria, we were able to see that hallmark structure, the granuloma form, nice tight aggregated macrophages, some infected, some not, and the occasional wandering neutrophils. So this was all good. Now, all of this makes the fish look big, but in fact, the fish is tiny and will fit on the cravat of Lincoln, as you will see here. So you have to devise ways to work with them. And this was developed by multiple people in the lab and perfected to an art form by Kevin Takaki in the lab. And what you'll see here is a vacuum suction device that takes the larva and latches onto it, and then pushes it to a hand-pulled glass needle that contains the bacteria with some phenol red that's used as a marker dye. And so let's watch them inject. And so that's the hindbrain ventricle cavity of the fish. And we can also bypass that and directly inject into the bloodstream of the fish. And if you're good at it, the way people in my lab are, you can do about 100 to 150 of these per hour. But oh my, didn't like that at all. But doesn't matter, we're done with that. So, so what you can then do is take these fish and array them into 96 well plates. And you can just put some fish water, it's just a bit of electrolytes in there. And the reason for that is that it's got this big fat yolk, so it doesn't need to eat for 2 or 3 weeks. So you don't have to feed these creatures. And this actually has great advantages because you can image the same plate every day. And what you can do is take that plate, stick it on ice. The fish will stop moving because of thermal anesthesia. And then you put them on the rotating stage of an inverted scope. You take the images. It takes 10 minutes to do that, by which time the fish will slowly start to move again. And you will get images like this. And we've been able to show that this relates very nicely to the number of live bacteria in them. So you can quantify infection. And you can do this every day. And if you're a nervous person, you can do them every hour. And it doesn't affect the fish badly at all. You can put drugs in here. and you can look at the effect of drugs without changing things. So it's this very small innovation that has really helped us to do the kinds of things that we've been able to do. Okay, so this is enter David Tobin, who came to my lab from Corey Bargman's lab, having done a screen for neurobehavior in worms. And he got interested in TB because he followed his wife to Guatemala for a couple of years. And while he taught biochemistry there at the university, he also went and attended TB clinics and AIDS clinics. And he saw that we had published a paper on this where we had mentioned the possibility of doing a screen. And he said, I'd like to come and do a forward genetic screen in the lab. So he comes to the lab, and we set up a forward genetic screen. And this is what we do is to simply make ENU, ethyl nitrosourea mutants, and we start to take these clutches and to screen them for susceptibility. And so here's an example of such a susceptible fish. Here's the wild type, and you can see this is a mutant sibling, and you can see that it is susceptible. But David made a very cool observation. He looked at these, and he had read the old literature, and he said that this looked like something called bacterial cording. Bacterial cording is a phenomenon discovered by Robert Koch when he first discovered the bacteria in 1881, where the bacteria grow in these serpentine cords owing to the presence of a surface lipid. And it's been used in the lab as a way to diagnose virulent mycobacteria, virulent tuberculosis. But what was very nice about this this connection that David made was that it then allowed him to do the mapping of the mutant by a binary trait rather than, I mean, this is less infection, more infection. That's easy to do to find your mutant in the beginning, but it's not as easy to do that when you're trying to map your mutation. And so, yes, no cording, yes cording is a binary trait, and that is always the best for genetics. And so David was able to use a more than century-old bacterial trait to do a eukaryotic mapping, and indeed he was able to show that cording mapped very nicely to 25% just about in the sibling in cross that was done here. Okay, so what the cording had revealed was that the bacteria had broken out of the cells, because within the cells, they can't really form these serpentine cords. And so what it told us was that the mutant had started to undergo necrosis in its granulomas. And we were able to show this in a more sophisticated fashion, or confirm it. There's our cellular granuloma, and there is the granuloma that's associated with very early cording. And you can already see that there's much less green, and you can see there's many, many more bacteria, and you can start to see that these little cords are starting to form wherever they can, and then it just gets worse from there. All right, so what was this mutant? The mutant mapped to the arachidonic acid pathway, to an enzyme called leukotriene A4 hydrolase, which catalyzes the conversion of an unstable epoxide leukotriene A4 to a highly stable, highly pro-inflammatory molecule called leukotriene B4. And here's our enzyme, and the enzyme was missing. And what David then realized by doing some biology was that the reason that the mutant was susceptible was not because it couldn't make this molecule. Because yes, this molecule is pro-inflammatory, and you surely need it if you're poked by a thorn or by for many other infections. But in the case of TB, this was dispensable. And the reason that the mutant was hypersusceptible was that in the absence of this enzyme, this substrate was being shunted to a highly anti-inflammatory lipid called lipoxin A4. And so there was a dominant anti-inflammatory effect that could not be overcome no matter how much of this you added back into the fish. All right, so David had shown that leukotriene A4 hydrolase mutants made too much lipoxin, and they had an anti-inflammatory effect, and particularly they couldn't make TNF, which is a critical protective factor against TB, and that resulted in uncontrolled growth. But at this point, we decided to stop and say, wait a minute, we are dealing with a phenotype in zebrafish, and we're not talking sushi here, we're talking caviar, because these are little eggs, not even adult fish. So does this, before we get carried away and take this any further, should we maybe look and see if this has any relevance in humans? And so it turned out that there's a common variant in humans that has that is in the human leukotriene A4 hydrolase gene, and you can either be a CC or a TT or a CT. And we showed that this variant mapped very nicely to levels in normals. So it controlled the expression of leukotriene A4 hydrolase. And then when we went to a cohort of TB patients, we saw that the data turned into a mess, because what our expectation was that the CCs would be susceptible and the CTs and the TTs would be resistant, because more inflammation should be good for TB. But it turned out that the results stubbornly showed us that it was the HETs that were protected, and the CCs and the TTs were both susceptible. We then have gone back to the fish and shown that this is true in the fish also, and we've figured out why. So I'm going to tell you this story now, and let's get started with the human data. So here's our promoter variant. It's 12 bases upstream of the transcriptional start site. You can see that it controls the level of LTA4H expression. It does it transcriptionally, and here you can see it does it for the protein as well. So now let's look at the human cohorts. So the cohort there are very few TB cohorts that have DNA or blood saved. And fortunately, Guy Thwaites in Vietnam had saved blood from his patients when he did a study in Vietnam that he published in 2004. And this cohort was a cohort of TB meningitis patients. TB meningitis is the most severe form of TB that is associated with as high as a 50 to 60% mortality, even when the organism is drug sensitive, and even when they're getting full antimicrobial therapy. When you look at the brains of individuals who die from it, it's not surprising. It causes a terrible basilar meningitis. And you can see that there's a lot of sort of white stuff here that looks for all the world like inflammation. So if David was right, if the zebrafish was a predictor, then we should see that, well, what should we see? We should see that the TTs would do worse. But in fact, what we saw was that the heterozygotes did the best, and the highs and the lows, the TTs and the CCs both did badly. And so let's pursue this a little bit longer, because what we began to think, based on what we had seen in the fish, was that the lows were dying from too little inflammation, and the highs were dying from too much inflammation. And it turned out that we were in a position to test this. The reason we were in a position to test this was because Guy had assembled this cohort to ask a question. And the question that he was asking was, does the addition of adjunctive glucocorticoid therapy, which is a broadly acting anti-inflammatory agent, does it improve the outcome, the dismal outcome of TB meningitis? And that was because I showed you that there was a lot of evidence of inflammation associated with it. And so the question was, is there a hyperinflammatory state that is causing increased deaths, and can we take that away if we add steroids? And indeed, everybody used steroids. When I went to Baroda Medical College, we used steroids with TB meningitis. But it was done on a physician dependent basis. And Guy wanted to do the study to see if this was warranted. So he did a placebo-controlled trial. There's dexamethasone, there's the placebo. And you can see that, yes, dexamethasone does do better, but very marginally. I mean, this is really not very impressive, is it? But because anything is better than nothing, at this point, dexamethasone became the standard of care. No longer physician dependent. You would have to do it if you wanted to practice good medicine. Now, this is what gave us the opportunity to test our model, because our model would be that maybe some people, the TTs were dying of too much inflammation, the CCs were dying of too little inflammation, as in, making less LTA4H and therefore too much lipoxin, which would be predicted by the fish. And so what do you think we might see if we now parse this by genotype? All right, let's have a look. So the placebos the hets do okay, placebo, dexamethasone. The hets do okay, no matter what you do with them. But look at the highs. 50% mortality in the placebo, not a single death if they got dexamethasone. So why then is this effect so low? It's because dexamethasone is actually killing people with the CC genotype or the low inflammatory genotype. And this is not acceptable because you're harming people to help a small subgroup of the population. So let me sum up this part. We went to the zebrafish, and the zebrafish come from the holy waters of the Ganges or the Ganga River. And I like to say that that's why they're transparent. And then we went to Vietnam, and we found that our variant did what it was predicted to do. And then because we had such an unusual association at the time, we then went to a cohort of leprosy sufferers in Nepal, and once again we showed that multibacillary leprosy was associated with either the CC or the TT, whereas paucibacillary leprosy was associated with the heterozygotes. And this is Guy Thwaites, who started collaborating with us way back then and has continued to do so. And Mary Claire King has been roped into TB from breast cancer and schizophrenia and deafness, and as has Tom Hahn and multiple people, physicians who work in Vietnam. So now the question was, why is it that high LTA4H is bad for you? Why is having too much of a pro-inflammatory lipid bad for you when you have TB? I can understand that it's bad for you when you have, heart disease or arthritis or colitis, but inflammation evolved to fight infection, so more should be better. Enter Fran Roca, so he then decided to pursue this question. So when you have a normal amount of LTA4H, you get sort of your normal amount of TNF, and that then allows the macrophages to control the bugs somewhat, and you get some expansion in the granuloma, as we've seen in other studies in my lab. Now, if you have too little TNF, then it's easy. That is the low LTA4H, the original variant. What happens is the macrophages can't control the bugs grow, and then the macrophages burst open, possibly because of a physical issue, but possibly because of some other pathway. The bugs come out, and now they have no controls, and they grow really well. What Fran showed was that if he overexpressed LTA4H in the fish, he was able to recapitulate what was seen in the human, as in he was able to show that these fish also were hypersusceptible with increased bacterial growth. But in the fish, he could go backwards and see what was happening. And what he was able to see was that the high LTA4H macrophages control the bacteria way better than the normals initially. And then somehow, suddenly, they popped open. And these few bacteria would come out, and then they would start growing. And very soon, you couldn't distinguish them from the low. So what might be the reason for this? So Fran showed that if TNF is in excess in the animal, when it binds to its receptor, it triggers the RIP1 and RIP3 kinases. And these, through a bunch of other proteins that I've spared you the names of, induces the mitochondrion of the infected macrophage to produce a lot of reactive oxygen species. Now, reactive oxygen species are normally produced, and to some extent, in response to infection. And TB has evolved to withstand that, so it doesn't care. But when reactive oxygen species are in excess, they can kill the bacteria, hence that finding of excess killing in the beginning. So I just want to show you, I'm going to show you a couple of bits of primary data because they're just so cool. You know, you can actually label live fish with a mitochondrially tagged dye that will light up only when there's reactive oxygen species. And you can see that at this stage, when there's high TNF, the infected macrophage is lighting up its mitochondria because it's got reactive oxygen species. And then you can see that shortly after that, because you can time all this, it's all live, and you can see with another dye that the macrophage is burst open. Well, Fran then showed that this was mediated by a matrix mitochondrial protein called cyclophilin D. And cyclophilin D is known to participate in the formation of a pore that is in the inner mitochondrial membrane. And when this pore is dysregulated and kept open, you lose voltage potential, and then the cell goes kaput. So we thought that this was the story because reactive oxygen species have been shown in the literature. Some might think that the literature is dubious, but what did we know? That reactive oxygen species are triggers for cyclophilin D translocation. So we thought the story was done. And then Fran did what my mentor Stan Falco used to call one-too-many experiments. And he did an unnecessary control. And he showed that for this pathway to work, this pathway that really could have been mitochondrion intrinsic and so nice and simple, needed the participation of lysosomal components. And it needed specifically the participation of an acid sphingomyelinase in the lysosome and its product, ceramide. So somehow these had to come together to produce necrosis. So what could that be? So Fran has since figured that out. And what he has shown is that when TNF binds its receptor, it triggers the RIP1 and RIPK kinases, and they produce reactive oxygen species. But what's happening now is a little different from what we initially thought. Reactive oxygen species are diffusing into the lysosome of that same cell and are triggering the activation of acid sphingomyelinase. Acid sphingomyelinase then it gets activated, produces ceramide. Ceramide then activates one of the most abundant lysosomal cathepsins, cathepsin D, and when cathepsin D is activated, it leaves the lysosome and makes its way into the cytosol. No organelle shall be spared today. So in the cytosol, cathepsin D comes out and activates a cell death pathway protein called BID. BID then activates a well-known effector of apoptotic death called Backs. And that appears to be the pathway. Now, I told you that Backs is thought to mediate apoptosis, and it's thought to do that by oligomerizing on the outer mitochondrial membrane. Cyclophilin D, which we knew was needed for this pathway, is involved in the inner mitochondrial membrane. And meanwhile, some papers came out that said that BACs can also mediate necrosis, which we were completely sure our pathway was necrosis, not apoptosis. And it said that BACs can mediate mitochondrial necrosis independent of oligomerization and in collaboration with cyclophilin D, and this was shown for heart conditions. So we said, great, TB is working just like heart conditions, and it's another example of a new role for BACs in the mitochondrial membrane, except that if that was the case, then you should be able to rescue a BACs mutant with a mitochondrially tagged BACs. So, just like bacteriologists, we also, when we do host genetics, we have to do due diligence. So for example, to show that each of these pathways are, each of these components are involved, you have to be able to add it back to the pathway and show that now you get back necrosis. So if you make a BACs mutant and say, oh, look, it no longer has TNF-mediated necrosis, you have to add back BACs and say, yes, it does have necrosis now. And so, of course, Fran had done that, but if these guys were right, as in if this pathway was working for us, then mitochondrially tagged BACs should be able to rescue necrosis just like untagged BACs could. But it didn't. It couldn't rescue it. Okay, so what was happening now? So what Fran then showed was that ER tagged, and I told you that we're going to get to another organelle soon. So he was able to show that BACs, which there's been all sorts of murmurs over the years. If you read the literature, anything can be involved anywhere in the cell death pathway. There's just, yeah. So anyway, people are nodding in a sympathetic fashion. So we knew that there'd been some rumblings about maybe BACs doing something at the ER. So BACs got tested at the ER, and Fran indeed was able to show that yes, ER-tagged BACs could rescue the loss of necrosis of the BACs mutant. And what he then went on to show was that what was happening was this. BACs was somehow causing the translocation of calcium from the ER. The ER is the cell's repository of calcium. And this calcium was going through the MCU, which is a uniporter for calcium to enter the mitochondrion. And that this calcium was entering the mitochondrion, and it was causing an excess of calcium, and that it was the calcium that was triggering the cyclophilin D to make its way to the inner membrane and cause necrosis. And again, I'll just pause for a minute to show you some nice live microscopy. So this is a transgenic fish that genetically expresses a mitochondrially tagged reporter that only fluoresces when the mitochondria have an excess of calcium, when it binds to calcium. How cool is that? It was made by my friend and colleague David Rabel at the University of Washington. So if you look at a fish that is just a control fish that's infected, you see hardly any labeling. And if you look at the TNF high or LTA4H high fish, you see lots of labeling. You can have a closer look at one of them, and you can see there's the macrophage. It's infected. You can see the label light up, and you can see that everything is on top of everything. And you can quantify this kind of thing, so you don't have to cherry pick your data. You can quantify this stuff blind, which is what we tend to do. And then he was able to show that not only was the BACs needed for death and for the calcium overload, but then he went back and showed that all of the upstream components, which we had already shown was needed for the death, was needed for the calcium overload. If you remove these components, not only did you lose the death, but you also lost the calcium overload. So this all looked pretty good. Okay, but how might BACs be targeting the ER to cause this calcium overload? And Fan ruled out that calcium was simply leaking out of the ER and going into the cytosol. He was able to show this genetically, and he was able to show that he was able to surmise that it had to be through a channel that went between the mitochondrion and the ER. And so what could that channel be? And the most likely was something called the IP3 receptor, because that's what's been associated with immune cells. Well, except that it turned out to be not the IP3 receptor, but another receptor called the Ryanodine receptor, which is associated more with the neuromuscular types of cells, with excitatory potentials and the transit of calcium for that. So he was able to show that Ryanodine receptor was the receptor that was involved. And he was able to also show that if he inhibited it with dantrolene, a well-known drug that's used in the clinic for neuromuscular emergencies, then you can revert the pathway back to wild type. Okay, so now I've shown you all this stuff. Anybody know who this is? Huh? Jeffrey Hall. So Jeffrey Hall won the Nobel Prize for circadian rhythms in 2017. And when they went to try to find him, they found him in his home somewhere in Vermont, where he had retired because he had retired early because he'd failed to get grants for like a decade and then gave up. So there he was in his Harley Davidson t-shirt, and at this interview, he said, the problem with all these modern people is that they're just, they just keep rattling off all these pathways and all these names, and he says, snore, authentic bio-gibberish. Now, but tell me what that thing does. Tell me why I should know about it, and then I'll wake up. And so that was Jeffrey Hall. So let me remind you why I care about this. So remember that I pointed out to you that while the highs did better with steroids, the steroids were actually making the lows worse. And so the idea was that's because steroids are broadly anti-inflammatory, so the lows are going to get worse. But what if you went down into the pathway that's only operant in the highs and got a pathway-specific drug like dantrolene? Then maybe things would be better. So we said, all right, let's have a look at that. And meanwhile, Guy had been trying to figure out if this repeats, because human genetic studies are fraught with not repeating. So he repeated this, and yes, to our great relief, it looked good again. Remember, he can no longer do the non-steroid arm, so we're stuck only looking at whether highs will do better than the lows. And then the study has also recently been repeated in Indonesia, so that and is ongoing in India. Okay, so it turns out that you can get lots of drugs from this pathway that are downstream of TNF and even downstream of reactive oxygen. One of them is disipramine. Disipramine is a tricyclic antidepressant that targets acid sphingomyelinase, it turns out. And that takes away the necrosis and relieves susceptibility, rescues susceptibility in the fish. We've got dantrolene for the Ryanodine receptor, and we've got a drug that's in clinical trials for cyclophilin D that they were kind enough to send us, and that too takes away the pathways. So we've got all of this. So this is all good. But Fran then had a brainwave. And he said, if all of this boils down to calcium. So he said, what if and one of the most commonly used drugs in the clinic are what? Calcium channel blockers. We use them for high blood pressure, we use them for migraines, we use them for cardiac arrhythmias. And so he said, what if we just reduce the amount of calcium going into the cell? And he went on to find that, in fact, the very receptors that these drugs tackle are present in macrophages. And he was able to show that, indeed, when he added verapamil, nifedipine, diltiazem, these are completely different classes of calcium channel blockers, that he was able to rescue the susceptibility. Okay, now, we get back to the age-old problem. It's like Camus, the plague, fascism you got to put down fascism all the time. So there's always going to be the complaint, is this just in the fish? So Meanwhile, my lab had taken to calling, everybody has names, right? We have necroptosis and pyroptosis and ferroptosis, and somebody in my lab decided that they were going to call his pathway rocatosis for Fran Roca. And Fran really liked this because he said calcium was involved, so it had a little, so it was really wonderful. And they've been playing with rocatosis, necrocatosis, and so on. So stay tuned for this to appear in the next Doug Green manual of cell death. So Laura Whitworth, who had actually done her PhD with Mary Claire King and arrived in my lab through a circuitous route, thankfully for me, and who's really mainly a human geneticist, was entrusted herself with the task of seeing if this would work in humans, this pathway. And so what we did was to take human macrophages, and I'll just walk you through this a little bit. So if this pathway is working in human macrophages, you have them on a dish, if you add TNF to them, they should die more. And indeed, they die more in a dose-dependent fashion, and that's shown very nicely here. And this is infected, uninfected from the same well. RIPK3 inhibitors, necrostatin 1, should remove the cell death, and indeed it does. Apoptosis inhibitors, which don't remove the death in the fish, should not, and indeed they don't. Then we can tackle all of these together. A BID inhibitor, there's a chemical inhibitor of BID, there's dantrolene, and there is a inhibitor of MCU called RU360. And once again, you can see high TNF gives you death. Death goes away when you add these inhibitors. And then finally, our sort of pièce de résistance pills. And yes, death with TNF, they go away completely with each of these 3 drugs. And then very finally, Laura got permission to use TB in macrophages so that we could now get away from marinum and the fish and do human macrophages with TB. And once again, she could show that here's the death with TNF, goes away with dantrolene, nifedipine, verapamil, diltiazem. So this was all good. So that was the day that their paper got accepted, and I pulled them out of the injection room here, and as you can see, they look pretty happy. So what's the take home here? Studying TB can uncover new biology, because remember that the idea that BACS would go and activate the Ryanodine receptor is completely new for a protein that's been studied for about 30 years now. And so if you let the bug take you places, maybe you can discover new things about the host. Similarly, this idea that organelles come together is now being more and more recognized in the context of homeostasis, but Jennifer Lippincott-Schwartz is a key advocate of that. But here we're showing that these organellar contacts actually can be pathogenic when in the context of infection. It exemplifies precision medicine because we've got drugs that can work for the high TNF state. We also, having said that, we actually are beginning to think now, based on other people's work, that a high TNF state locally within necrotic granulomas may not only be determined by LTA4H, but there may be many other reasons that will lead to a high TNF state, and this may then be a way that you can treat all of them. And we've got safe and inexpensive drugs that are that could be repurposed, FDA approved, so this is a good thing. And we're particularly interested in the drug verapamil for the reason that many years ago, Kristen Adams, when she was a grad student in my lab, discovered that bacteria induce efflux pumps. We're pivoting to bacteria. Mycobacteria, TB, induces efflux pumps when it gets into a macrophage. These efflux pumps allow the bugs to survive. They're virulence factors. But they also then kick out antibiotics. And in particular, she had shown rifampicin, isoniazid, you name it. Every antimicrobial against TB is subject to macrophage-induced efflux. And she had gone on to show that verapamil, that if she used verapamil, she could inhibit this efflux. And so we are now trying, there's a glacially paced trial of verapamil as an adjunctive therapy for TB that's made its way to phase 2 in India. And now we can say that verapamil could have a dual action, a double whammy, by also targeting a host gene. Okay, you've probably forgotten every pathway I mentioned, just like you know, our snored bio-gibberish hero. And so I'm going to tell you the message in a way that might make sense to you. So this is a river in the United States. It's not the Ganges. Does anyone know what river it might be? So, okay. It's a river on the, it's in Yellowstone National Park. Yep. And it's the place everyone goes to after hot days backpacking or hiking. And they got so tired of people telling people not to go in that they finally just made a way to enter. So you kind of walk down this way, and then you get to a place and you can enter. And as you're walking along, we're walking along here, our family, and my husband says, hey, look at all these people. He says, this is your pathway. And well, we finally understood what he meant. And if you go in, you look and you see, you really see that everybody's in one line. And why might that be? This is a hot spring feeding into a cold mountain river. If you go, literally go here, if you go even one bit here, you will boil. And if you go here, you will freeze. You can only be comfortable in this one line. So I went to the Convent of Jesus and Mary in Baroda, and that's how we had to walk everywhere to recess and everything, or we got, you know, caned. So, okay, and so this is, if you're here, you do well with TB genetically. If you're here, you do badly, and if you're here, you do badly. But if we know this, we might be able to help, because we've got drugs now to bring you back from the hot to the middle. And in a story that I'm not going to tell you today, we are now beginning to figure out the low LTA4H and how that might work. And that is leading us to another cheap little drug called ibuprofen, which some of you might even have in your pockets right now. Thank you. No, yeah, I can take questions if they want to.
So I think we have time for a couple of questions. Actually, well, when you bring the microphone over here, because this one is tainted now since I grabbed it.
TB.
Right over here.
Lali, I wanted to know your thoughts about these therapies for non-CNS.
Keep the mic closer, yeah.
Non-CNS TB. Is there an effort to look at, you know, how this scheme works also for pulmonary tuberculosis?
Yes.
And can you comment on
so,
that?
Guy, so, you know, it's a little harder to do that. So the question is, what about pulmonary TB? And we are interested in doing that. So Guy has a study going to look at pulmonary TB. Now remember, drug-sensitive pulmonary TB in a center like Guy's will have very little mortality. So you can't do it a mortality screen. So what they're doing is whether people clear earlier or whether their X-ray findings revert earlier, and also whether they're more susceptible to begin with. So that's going on in Vietnam, and is also, it's happening at a good clip in Vietnam and at a very slow pace in India and Indonesia.
Shorten the duration of therapy, which is a major problem.
Yep, we would like all that, but you know, we should be very careful. Yeah, but I don't want to overstate the case. You know, I feel like we've been very lucky with this. You know, it's sort of remarkable. But those are the kinds of things we're doing. Yeah.
You go first.
Yeah.
Lali, so it's my understanding that MLKL, which is the downstream target of RIP3, is missing in vertebrate fish, including zebrafish. And so I'm curious to know what the downstream, like the business end of necroptosis is.
But this is not necroptosis.
Well,
Remember? Wait. It's not necroptosis because it's going through the mitochondrion. That's not.
But it's RIP3 dependent.
This is a completely different pathway. This defies any kind of categorization. Because this is neither, you know, if you look at Doug Green and, you know, all the gurus of cell death, they had cyclophilin D mediated, mitochondrial cyclophilin D mediated death. That is not TNF. Then they have TNF mediated necroptosis. That is not the mitochondrion. And this is neither of the above. This is something that the bug is inducing, because the bug is needed in this pathway.
So then, do we understand
And as for your question about MLKL1, which is this protein that's involved in necroptosis, which this is not, fish don't have MLKL. However, if you use necrosulfonamide, the agent that was initially found as a target to target MLKL, the pathway is rescued. However, we now know that necrosulfonamide also binds
targets
gasdermin.
Right.
So, you know, we don't know what that's doing.
Okay.
But I think you've got to stop. You can't think of this as necroptosis. It is not.
But the ultimate target that's killing the cell is not, right? We don't know what that is in this case.
We do know what it is. It's cyclophilin D. Cyclophilin D is, if cyclophilin D starts to dysregulate the pore, the transition complex, you're getting a loss of voltage potential, and you're dying of that.
Okay.
Yeah, we very much know that. Yeah, it's entirely. The targets are calcium, cyclophilin D. So if you want to think of it simply, think TNF, reactive oxygen species, big mess, calcium, cyclophilin D. Yeah. Oh, sorry.
Over here. So one of the big disappointments from the MCU field is that the mice are viable, right?
Pardon? What?
The MCU knockout mice are viable.
Yes.
Do you think that it's because it's actually an immune?
The MCU knockout mice are viable.
Yeah.
Okay, and
so in your pathway, they would no longer be able to perform the the rokatosis.
the MCU knockout mice are viable with TB, you mean?
No, I don't know how. Has anyone tried it? That's.
I have no idea. We tend to bypass the mouse and go directly from fish to man. And then they looked from fish to man and man to fish, and none could tell which was which.
So it would be, I guess it would be interesting to see whether those MCU mice, because
yes, but you remember, you'd have to create a high TNF state, because this pathway is not going to start unless you have the trigger upstream. You see what I mean?
But yes.
You'd have to get Raphael to inject those mice every day with TNF.
Or just give them TB.
No. Well, yes, maybe.
Sorry.
I was excited about this new role of BACS with the ER. So I was wondering
new role, what role?
BACS with
yes.
the association with the ER.
Yeah.
And I was wondering if you thought that there was a mechanism to prevent it from oligomerizing and going through the canonical path.
So remember, this is non-oligomerization dependent.
So is there a mechanism to prevent it so that it shouldn't oligomerize, so that it knows that it has to associate with the surface of the ER? Like, is something happening for it to not oligomerize?
Yeah, you know, your question, underlying your question is a sort of, to me, the main puzzle here. Because why, I mean, the thing I can't quite understand is why isn't BACS doing what
it's supposed to do?
Going to the
exactly.
mitochondria,
That was
right?
my question.
And it's got to be a kinetic thing. Because there's nothing, I don't think there's anything actually preventing it. Now, it could be that there's some caspase 8 that's doing something different, but I think the main issues here are there's got to be things that pathways, I think they're all kinetic changes that push molecules to go in one direction or another. And those are the kinds of things that I think are going to be, I think those are going to be really hard to figure out. I mean, I think we're going to be able to figure out how BACS binds to the ryanodine receptor, you know, or what it activates. Maybe there's an intermediate. That part, I think, we'll be able to solve. But this idea of what causes pathways to go in one direction versus another, and that's not just for this. I think I'm starting to see that problem in all of our field. I think that's what we're all going to be grappling with.
Great, thank you.
Okay, just for the sake of time, okay, we'll do just 2 more questions. So.
mechanism you showed connecting all the dots
Yeah.
is really beautiful,
Yeah.
but can you comment on what the role of the actual intracellular infection has?
The intracellular
The
what?
infection itself,
Yes,
because
I
if you
can-
add TNF and you
I can-
follow the
I
dots.
can tell you the limit of our knowledge about it. That's going to be our next question. So what we do know is that it's upstream. So without the, you need TNF, you need excess TNF signaling and the bug in the same macrophage in order to get the pathway going. And you need it right at the level of reactive oxygen species. So if you don't have an infected macrophage, then you can signal with as much TNF as you like. You're not going to get the pathway to go. And in the fish, you can do this very nicely because the neighboring macrophage that is infected will trigger the pathway, but not the macrophage that is uninfected. What we don't know yet, what I can't tell you yet is if that's the roadblock or whether infection is also doing something downstream. And we've thought of some tricks to figure that out. So, and then we'll know. But what we'd really like to go after is what is the host, what is the bacterial factor?
Okay, one last one over there.
In the human studies, do you think that it's just this SNP? Because it was very clean. The
I know.
lows were all surviving. And can it really be just mono one gene?
Well, so let me tell you a little bit more about this variant. This variant is curious because its distribution in the world is curious. It is the most common, it's common in broadly Southeast in Asia, actually all of Asia. So, and its frequency is about 35 to 40%. Africa is in the middle at about 12%, and Europeans, it's very low, about 3%. And this is odd for a distribution because Africa should always is normally at one end, not in the middle. So, it obviously evolved in a curious way, and as to why it evolved that way, we don't know. And in this population, there's it seems to be controlling it because the data are very clean, and they've come out very clean again. And that's why I was saying to whoever it was that we just got incredibly lucky. They're so clean that we ourselves were suspicious at the beginning, but we've now, verified it in 4 cohorts. And whether whether in Europeans there's going to be some other variant controlling it is something we need to look at. Very difficult to do in Europeans. You could go to places like Russia maybe, because you have to go where there's enough TB to do the experiments.
Okay, thank you. Let's give Lalita a round of applause.